This is Gladstone Audio News, where you'll hear audio versions of the latest stories featured on gladstone.org. These episodes are narrated by an AI voice for accessibility and convenience. Inside the Brain, Tackling Neurological Disease at its Roots. Published on July 21, 2025, on gladstone.org. From discovering therapies that can repair the brain after stroke to using machine learning to detect early signs of cognitive decline, researchers at Gladstone Institutes are at the forefront of neurological research. Our scientists are not only working to uncover and understand the biological mechanisms behind conditions like Alzheimer's, Parkinson's, brain injuries, and more, but are also striving to turn their discoveries into treatments for patients. On World Brain Day, we're spotlighting groundbreaking research from Gladstone that expands our understanding of the brain and brings new hope for treating neurological diseases. 1. Stem cell therapy jumpstarts brain recovery after stroke. Gladstone researchers, led by John Paz, Ph.D., worked with a team at the regenerative medicine company SanBio to show that a cell therapy derived from stem cells can restore normal patterns of brain activity after a stroke. While most stroke treatments must be administered in the immediate hours after a stroke to have a benefit, the study, published in Molecular Therapy, showed the therapy was effective in rats even when given 1 month later. 2. Brain immune cells amplify damage caused by Alzheimer's risk gene. A study led by Yadong Huang, MD, PhD, reveals how the APOE4 protein, known as the strongest genetic risk factor for Alzheimer's disease, interacts with immune cells to drive neurodegeneration. For this study, published in Cell Stem Cell, the researchers used an innovative model in which human neurons producing APOE4 were transplanted into mouse brains. They found that when they removed microglia, immune cells in the brain, the APOE4 protein no longer triggered as many deposits of amyloid or tau, two types of misfolded proteins that are hallmarks of Alzheimer's disease. This study highlights a promising new target for Alzheimer's therapies. 3. Preventing brain damage in preterm babies. Visiting scientist Mark Peterson, MD, worked closely with the lab of Katerina Akasoglu, PhD, to identify a root cause of developmental delays that result from brain bleeds in premature infants, opening a path for new therapeutic strategies to counter long-term health risks. In a study published in Proceedings of the National Academy of Sciences, the team showed for the first time that a blood protein called fibrin blocks an essential biological process that drives brain development in early life. 4. Machine learning reveals behaviors linked with early Alzheimer's and points to new treatments. In a study published in Cell Reports, a team of scientists, including Stephanie Miller, Ph.D., and Jorge Palop, Ph.D., used a new video-based machine learning tool called VAME to pinpoint otherwise undetectable signs of early disease in mice that were engineered to mimic key aspects of Alzheimer's. The open-source tool identified subtle behavioral patterns captured on camera, changes that might not be noticed by simply looking at the mice. This work sheds light on a new strategy for identifying neurological disease earlier than currently possible and tracking how it develops over time. 5. Discovery of how blood clots harm brain and body in COVID-19 points to new therapy. A study published in Nature and led by Katerina Akasoglu, Ph.D., and Warner Green, M.D., Ph.D., revealed that the blood protein fibrin is not just a byproduct of inflammation in COVID-19, but a primary driver of the disease's clotting, inflammation, and neurological symptoms. The research shows that fibrin becomes abnormally toxic during infection, binding to the virus and immune cells to form clots that trigger widespread damage and suppress the body's ability to clear the virus. Importantly, the team also identified a promising antibody therapy that neutralizes fibrin's harmful effects, offering a potential path to treating both acute and long COVID. 6. Powerful new tool can identify cells promoting health or disease. Stemming from her work on Alzheimer's disease in the lab of Leonard Mucha, Ph.D., Catherine Sant collaborated with Ryan Corsis, Ph.D., to develop a computational tool called Quire for distinguishing cells in complex biological samples. The study, published in Nature Genetics, shows that Quire outperforms existing methods by accurately identifying biologically meaningful cell types and cell states without generating misleading results, offering a more precise way to uncover cellular drivers of health and disease. 7. A new therapeutic target for traumatic brain injury. Gladstone scientists, led by J.Q. Ryu, Ph.D., a scientific program leader in the lab of Katerina Akasoglu, revealed that fibrin, a blood coagulation protein, is responsible for initiating toxic inflammation and neuron loss after a major head injury. The findings, published in the Journal of Neuroinflammation, can inform new treatment strategies for a condition that often presents long-term health challenges. 8. Cellular Clean Energy. Can mitochondria make more energy without collateral damage? A team at Gladstone led by Ken Nakamura, MD, PhD, used the gene editing technology CRISPR to investigate within mitochondria exactly which molecules are responsible for creating energy versus those that control the production of reactive oxygen species, or ROS, toxic byproducts commonly known as free radicals. Since impaired mitochondria are believed to play a central role in a host of neurodegenerative diseases, the findings published in Proceedings of the National Academy of Sciences could lead to the development of therapies for diseases such as Parkinson's or Alzheimer's. 9. Combination of two cancer drugs may work against Alzheimer's. Scientists at Gladstone Institutes and UC San Francisco identified cancer drugs that show promise for reversing changes that occur in the brain during Alzheimer's disease. In a study published in Cell, Yadong Huang, MD, PhD, and his collaborators used public data to identify gene expressions associated with Alzheimer's, then looked at data from more than 1,300 drugs that change those gene expression signatures. They found that when tested in mice, a pairing of two FDA-approved drugs reversed multiple aspects of Alzheimer's and restored memory. This was Inside the Brain, Tackling Neurological Disease at Its Roots, published on July 21, 2025. Visit gladstone.org to learn more about the groundbreaking research conducted at Gladstone Institutes.